Thank you, Simone, and thanks for inviting us. And it's great to hear that we'll be sharing the slides, because the thing that I've noticed while looking at the slide that's in front of us now is the danger of recycling, because this is a slide I've used before, and I see it's only got my name on it. It hasn't got Stefania's name on it, which we will correct when we share these slides with you all afterwards. So I apologize for that, Stefania. And Stefania's been too kind to correct these slides while we've been circulating them. So as Simone said, we're going to talk about the good, the bad, and the ugly. This piece of work that Stefania, myself, and some colleagues who I will mention later on did last year, looking at the proportion of trials that are bad. That was our main aim. And this actually came about because of some work that was prompted by Declan Devan inviting me to give a talk in 2017. You can see the dates on this slide here. And it was a talk to systematic reviewers. And Declan had asked me to talk about, what do you think systematic reviewers should be thinking about? And I ended up coming up with this talk, which was called something like Why Cochrane Reviewers Should Start to Get Stroppy About Poor Quality Research. And what I did for that, as part of it anyway, was go onto the Cochrane website and look at, as you see on the right, it says latest Cochrane evidence. I just pulled a few of those to see what proportion of the trials included in those reviews were high risk of bias, low risk of bias, uncertain risk of bias. And hey presto, what it showed was that most of the trials included in the systematic reviews were high risk of bias. In fact, there was one review that was over 1,000 pages long, had more than 200 trials in it, and every single one was high risk of bias. So that was a device that I've used a few times for other talks. So I've done exactly the same, different year. Let's try it again, look at another 4 or 5 reviews. It's the same result. It was always the majority of the trials within those systematic reviews were at high and or uncertain risk of bias. So it either had very high proportions of high risk of bias or remarkably high proportions of uncertain risk of bias. And that had been sort of kicking about in my head for a while. And I thought, wouldn't it be nice if we could actually do it properly? So instead of me just picking 4 or 5, why don't we actually look at a massive body of reviews and do the same exercise, but now for hundreds of reviews, say, and including hundreds and hundreds of trials. So that's where Stefania came in, and we'll hear from Stefania in a moment. So what we wanted to do was to look across a body of reviews, look at the trials, and ask that same question: what proportion are at high risk of bias, what are at low risk of bias, and what are at uncertain risk of bias? And as you can see, that's Stefania, who did the vast majority of the work linked to getting hold of all the data that we'll be talking about in a moment. And we also worked together with Francis Sheely at Cork and Mike Clark in Belfast. So it was a nice collaboration between TrialForge and the Health Research Board Trial Methodology Research Network. So a great piece of work to be talking to you about because the TMRM was such an important part of the work. Okay, and at this point, I'm going to hand over to Stefania to talk about a few of the slides, and we'll do a bit of to and fro for the next few. So Stefania, please.
Thanks, Sean. Hi everyone, and thanks for joining. So on to my piece of work for this project. What we did is that we randomized 2 reviews from each Cochrane Library Review Group between May 2020 and April 2021. I read every single review and extracted basic information from every single review, like number of reviewers, number of participants, intervention types, the control groups, outcomes, and so on. But most importantly, I extracted the overall risk of bias for each trial included in each randomized review. And we called the high risk of bias trials bad, the low risk of bias trials good, and the uncertain trials the ugly trials. And then we use the R program to look at all this collected data and analyze it and see what's going on. So Sean, if you go to the next slide, please. So in terms of scale, we sort of wanted to look at the percentage of trials that are bad to see whether or not Altman's words, so Altman's words were true more than 2 decades later, whether most of the trials are bad. And then we calculated the number of participants that are involved in those bad trials. And we tried to come up with an estimate of how much would the bad trials cost based on the cost per participant, which Sean can talk about now.
There we are. As Stefania said, we were dead keen on trying to be able to say something about how much money is spent on trials that are bad, in other words, high risk of bias. And because we had the number of participants that Stefania was extracting for each included trial, that gave us an opportunity to do that if we could calculate a cost per participant. And what we wanted to do was get a few estimates. And so we didn't want to make them up ourselves. We wanted to look to the literature to see if we could find a number or whether we could calculate a number. And we actually came up with 3 estimates, all based on published data. So we didn't make up the numbers ourselves. We obtained them from published data. So the first one is an estimate that comes from the UK's National Institute for Health and Care Research Health Technology Assessment Programme. And what we did there was essentially work out the cost per participant by getting the average cost of a trial and the average number of participants in an NIHR HTA trial. And that came out at just under £3,000 per participant. Okay, so that was one estimate. Okay, well, let's look for some others. So we also found a US paper that actually gave a cost per participant. This one's a median cost per participant that they gave of about $41,500. And that was for trials supporting approval of new drugs, essentially. So a different type of estimate there. It's quite a bit bigger. And it was for a particular type of trial. So these licensing of drug trials. And then we found a number, which I was already familiar with, this last one, which is a fairly old average cost per participant that was published, I think actually in the BMJ in 2012 or thereabouts, which was just under €10,000 per participant. So what we did with these numbers, as you can see, they're in 3 currencies, they were done in 3 years, is we basically brought them all to the same year. So we looked at the exchange rates, and then we did a translation conversion rather from the currency that they're in, if it's dollars or euros, to GB pounds. So we converted everything to 2021 pound sterling in August. That was what we did. And there's a couple of websites we used for that, which are described in a paper that is linked to this work. But we brought them all up to the same level, if you like, 2021 sterling. And there are 3 estimates that we then thought, okay, what we'll do with the cost is we'll apply each of these estimates and we can see what the range is. Okay, so Stefania, you're back now.
Yeah, so the slide is really to show you the scale of the work that we've done. And there were 96 reviews included from 49 clinical review groups with a total of 546 reviewers. And out of those reviews, we extracted 1,659 trials, like we said in the beginning. And to add on to this slide, I just wanted to say that there were also 84 countries involved in those extracted trials. 193 of those trials were actually multinational. And I'll show you a figure on the next slide on the clinical groups. So here you can see the risk of bias per clinical group. And well, if everything was perfect, then what we would see would be a solid block of purple, but we don't see that. unfortunately. And we can see here that anesthesia is doing the best, apparently, as a clinical group. It's got the highest percentage of good trials. We don't know why that may be. We can only assume that may be because they must have better methodological or statistical support. But also what I found when we did this research and I took a closer look at the data, we noticed that drug trials generally do better than non-drug trials. Again, we don't know why that may be. Might be because drug trials are generally better regulated. And anesthesia, obviously, it speaks by itself. These are all drug trials. So I assume that may be why it's got more good trials than the rest, but we don't know. And also I wanted to add, in case you're wondering how the UK is doing about the good trials, actually 11% of the UK trials were good, and best trials were multinational trials. I think the percentage was a bit over 20% of good trials. So yeah, Sean, do you want to move to the next slide, please? So out of the 1,600 trials that I was just talking about, unfortunately, 62% are bad, 30% are ugly, and 8% only are good. And as you can imagine, most of the participants that are recruited for those trials are included in those bad trials. And as someone who's new to the field, because I started working with Sean in my first year of university, this was sort of shocking for me, because these are patients who are at stake for the bad trials that many people are conducting. And this for me is, like I said, it's shocking. And when Sean and I first started working together, our first project was on recommendations from Cochrane systematic reviewers. And I remember two of their main recommendations were the larger sample size needed for trials and the longer follow-ups needed for trials. And as a science student, that was, again, very shocking for me, because the first thing they teach us in university is that in order for us to get significant results, we need a large sample size. And it was shocking, because grown-up scientists don't follow their own advice. And again, because I am new to this field, for me, what's most enraging actually is that those patients who sign up for those trials hope for something better to happen to them, and it's not. Yeah, so I just wanted to bring my own opinion into this.
Okay, so if we go to the next one, I think this one's back to me. And as Stefania was saying there, that thought about the number of participants who are in these bad, high risk of bias trials, that's what this slide is showing. And as Stefania just said, nobody goes into a trial as a participant expecting that trial to be of low quality. Everybody is giving their time and their goodwill and their participation because they have a belief that this trial will help science, it will help people like them and perhaps themselves. And what this slide shows is when we extracted the data that Stefania had got for us, and we divided the trials into the good, the bad, and the uncertain. And what we find is the majority, 56% more or less, of participants, well over 200,000 of the 400,000 that were in this group of trials, more than 200,000 participants find themselves in a bad trial. And actually, it's interesting, I think, here to note that well over 100,000 are in trials where we don't know whether they're good or bad, so they're uncertain. So a very large proportion of participants are in either a bad trial, or what Mike Clark called a maybe bad trial. It's almost 90% of participants. So that is one of the most important points. All of these people who are giving their time and participation, taking part in the trials, have an expectation that those trials are going to be of high quality, and that is not the case. The majority are not, as we have shown here with these numbers. Okay, so if we move on to money now, we were quite keen on being able to say something about scale, as you'll remember, something about the number of participants, then also what about money? And we had those three estimates that I talked about a moment ago. And if we were to take those low and high estimates, then the high risk of bias trials, this is just the high risk of bias trials, then the cost of those trials is between 726 million and 8 billion. And remember, this is just these trials in our cohort, the sort of, I think it's 1,013 high risk of bias trials in our cohort. This is not global spending. This is just spending in that cohort of trials based used on the estimates that we got. So very, very large numbers. And what, again, to sort of put it into some sort of perspective, we also looked at what could you spend that money on? And we used the National Institute for Health and Care Research Health Technology Assessment Programme, so the UK's biggest really public funding stream for trials. And if we had that amount of money that's on screen right now available to us, that at the low end, 726 million, that would fund the UK's biggest public programme of trial funding for just under a decade. And at the high end, it would fund the NIHR for over a century. Like we'd be in clover, like we've got 100 years of funding there. And again, something to highlight here is this is not global, like total global activity. This is just within those 1,000 trials in our cohort that were classified by Cochrane reviewers as being at high risk of bias and using our estimates. It's a barn door of a range there, 726 million to 8 billion, massive. But whichever way you look at it, it's a lot of money. And I guess none of us would want to be seeing money spent on trials where we have serious doubt as to whether we can believe the results of the trials. And that's the point we were trying to make here. It's a lot of money. We could argue about the numbers, we could argue about the estimates, but it's a lot of money. And that is not easy to move away from. It's a lot of money. Now, Stefani, when we were doing the project, was very keen to say, surely there's something nice we can say out of this, something positive. And as Stefani mentioned a moment ago, there were some pointers towards differences that which suggested something more positive. And this is linked to drug trials, this particular point that's on screen right now. And it's looking at the low risk and the high risk. And for drug trials compared to non-drug trials, the proportion of low risk trials that are drug trials is higher, and the proportion of drug trials that are at high risk is lower. So again, as Stefani said a moment ago, there is a suggestion there that drug trials are a bit better than non-drug trials. We don't know why, and we can speculate, and perhaps there'll be interest in discussing that later. But this is what we saw in the data. There's some suggestion that drug trials are a bit better than non-drug trials when it comes to risk of bias assessment. And another thing we were interested in was, how is the risk of bias changing with time? So we know about tools such as the Cochrane Risk of Bias tool, and we were wondering, well, what sort of impact does the knowledge of that tool, if you like, have on trialists and how they design trials? Does it make any difference at all? And we looked at the date of the trials that were included within our cohort, and then looked at how risk of bias, the proportion of low and high uncertain, changes. And actually, there is a bit of a positive story, which is that the proportion of trials that are low risk of bias is increasing with time. So if you were to plot a graph of year against low risk of bias, that proportion is going up. The problem is it is going up very slowly. So if there was no impetus pushed into that system to try and get that rate to speed up, but it bowled along at the rate that it's increasing right now, then all trials would be low risk of bias by about the middle of the 22nd century. So it's very slow. It is improving, but it's very slow. And that suggests to us is that, really, we don't want to wait until Aberdeen looks like this particular picture. Well, that's when we're going to be satisfied that that's brilliant. All trials are now low risk of bias. We would like that to come sooner. There's no real excuse for it not coming sooner. So we thought what we would do is try and give some recommendations. So we've got this idea about scale, we've got this idea about participants, money. There are some signs of progress, but none of it's fast. So we thought we would make some recommendations, and we made 5. And we deliberately wanted a small number of recommendations. And we're not really claiming any originality with these. So many other groups have made some similar sorts of recommendations to these. What we wanted to do was present a small number, which we thought might have a fairly quick effect if they were implemented, which matched with what other people had suggested. So we're not disagreeing with anybody, but which come from, if you like, some of the problems that we've highlighted in the quantitative work that we presented about scale and participants and cost. So the first one of our recommendations was, I guess it's basically astonishing to us that so many trials happen when that come out at high risk of bias. So presumably, most of these are going to need funding from somewhere. We don't do trials for nothing. So some that will need funding. And our recommendation was, well, look, we shouldn't fund any trial unless the trial team has methodological and statistical expertise. And there are bits of work done by others that have looked at trials that have been high risk of bias and looked to see how easy or difficult it would have been to fix those problems. And some of them, and this is quite depressing really, some of them are actually really quite simple to fix. So the only reason they and we could think for those problems not being fixed, because they were simple to fix, but they weren't fixed, was because there was nobody in the team who recognized that it was a problem. In other words, they didn't have the skills and experience and knowledge to do the trial well, because they lacked methodological and statistical expertise. So our first recommendation, again, it's not original, others have made the same one, is look, trial teams need to have methodological and statistical expertise. So if the trial funding panel doesn't see somebody who is a professor of biomedical statistics or something like that, then you'd ask a question, well, who is your statistician? What expertise do they have? Similarly, who's providing methodological input? Recommendation 2 is actually quite similar. One of the points that Doug Altman made in his 1994 paper, The Scandal of Poor Medical Research, was that the reason we have the problem with poor research is that people who lack the expertise and skills, experience to do trials still manage to do them. Nobody stops them. And ethical approval is a stage at which to stop a bad trial happening. We'd all have expected it to have been stopped earlier, but it's another opportunity for a bad trial to be stopped. So we thought, right, ethics is a place to stop them. If they've managed to get through funding, they'll all need ethical approval. So the ethics committee asks the same question: who is the methodologist? Who's the person with statistical expertise? And if there's no obvious answer, then you don't give it ethical approval. And what we'd suggested in the paper, the suggestion of Frances Shealy, what she was thinking of was the very large number of participants who had been involved in trials which were high risk of bias, these bad trials. So she thought that would be a sensible question for the public and patient contributors on ethics committees to ask as part of their role, if you like, of protecting the interests of future participants, is to ask, who is the person or persons who are providing statistical and methodological expertise in this trial? How can we feel reassured that this trial is going to be low risk of bias? The third point is cheap as chips to implement, is let's use a risk of bias tool at trial design, not only at the end. So risk of bias tools were developed principally to support people synthesizing evidence of systematic reviews, by which time it's too late. The damage has been done if you're basically sort of just looking at the trial and saying it was bad, or look, it's great. But there's nothing to stop you applying those sorts of tools at the design stage and looking at your randomization and your allocation, for example, and making a decision. Okay, actually, I think we've got a bit of a problem here. Maybe we would change that because we think our current approach to, let's say, allocation is likely to introduce bias. So let's change it. So you use it at the design stage, and those tools are available. Some of them are quite simple, some of them more complicated. We would, I guess, be suggesting the simpler ones. But it's a tool that is there. There are tools. You could use it at the trial design, make changes, and avoid these problems further down the line when actually there's nothing you can do by that stage. You've just got a poor trial. Now, all of those above things, and particularly the first two, where we're calling for methodological and statistical expertise, only works if we have methodological and statistical expertise available. So our fourth recommendation was, look, we need to have more methodologists and statisticians. And this is definitely a call that people have made before. One of the problems with teams running trials or designing trials without access to, or without the involvement of statisticians and methodologists, because there aren't any around them. They can't find them, can't get hold of them. So we think it would be useful to basically train and support more methodologists and statisticians. And the last point is, well, we think there needs to be an infrastructure, if you like, put around methodology and the infrastructure to support trialists in doing high quality trials. And one of the points we made in our article was money's always tight, but as we saw from those numbers, there are some very eye-watering amounts of money being spent on trials which were classified as very high risk of bias, bad in our terminology. And perhaps some of that money would be better put into improving infrastructure to support trialists with regard to statistics and methodology. Because our view would be that all or most of that money, if you're putting it into a trial which is bad, is less likely or indeed unlikely to produce results which you can trust and use, and better to do fewer but better trials. So if we come on towards the summary now, what we're saying in this article and through all the data that Stefania extracted and presented to you earlier on is that if we have little or no confidence in a trial's results, then that whole trial has been a waste. And that was Doug Altman's point in 1994, and it remains the scandal that it was in 1994. If you look for trials that are high risk of bias, you will find them. Most trials are at high risk of bias if you go out there and look for them. And that's what I found in my half-baked approach, just looking at a few off the Cochrane website in readiness for a presentation. It's what we found in this much more substantial piece of work that Stefania helped us with. And we think we should be putting rigor and methodology exactly where it begins, right at the center. Like it's absolutely central. There's no point having all the other stuff unless you have that rigor and appropriate methodology right at the center of it. There's plenty of opportunity to waste an awful lot of people's time and goodwill and, of course, money. And we think the recommendations we gave are not a bad place to start when it comes to thinking about ways to reduce the proportion of trials that are high risk of bias. And I'll end with this comment from Doug Altman, since it was the scandal of poor medical research that kicked off some of this work, is that he, in the article, made this comment, is that we actually need less research. We don't need more, we need less. But what we do need is better research, and we need research done for the right reasons. And that better research bit in particular, I think, fits very nicely with what we're talking about. We think the majority of those trials that fall into our bad category, the world could have not had them and would not have lost very much. I guess that's what we're saying. So we want better research so that we don't find ourselves in that situation where we have a trial we don't trust its results, and therefore we can't do anything with it. And yet all of that resource, time, goodwill has been wasted. Okay, so I'm going to stop there, but I wondered, Stefania, if you had any additional thoughts you wanted to add before we start perhaps taking questions, if there are any.
Actually, as you were talking, Sean, I was thinking, this is just a thought that I had. It would be really nice also if students who go into medicine and or go into sciences, it would be really nice if us as students would be taught more about the importance of methodology and stats in our studies, because I don't think enough importance is put on this. And indeed, there should be specialized people in methodology and statistics, but at the same time, everyone who's involved in those trials should be aware of the importance of methodology and stats. And a good place to start would be during your education, right? Just a thought that I had. But yeah, I can see some questions in the chat, really interesting questions.
Okay, thank you. Good point to end on. Basically, teaching people from the very start of their careers would be a good idea. I'll stop sharing, Simone, and then we can perhaps try and answer some of the questions.